That's a bit about Penguins. Study 2019, number 2, population density, osmosis, and experimental design. The student studying two different aquatic plant-eating unicellular protist species, species A and B, designed an experiment to investigate the ecological relationship between the two species. In treatment group 1, the student placed 10 individuals of species A into a container with liquid growth media and 10 individuals of species B into a separate container with the equal amount of the same liquid growth media. In treatment group 2, the student placed 5 individuals of each species in a single container with the liquid growth medium. The student then maintained the conditions under the same environmental conditions and recorded the number of individuals in each population at various time points. The results are shown in Table 2. So here we can see Table 2. We can see their population growth, or how much we had of that number of individuals in group 1 when they were separate versus group 2, which is when they were together. So the growth curve of species B in group 1 and the species A in group 2, the shade columns, have been plotted on the template. Use the template to complete an appropriately labeled line graph to illustrate the growth of species A in treatment group 1 and species B in treatment group 2, the unshaded columns. So first, let's figure out what's on our axis. So on our x-axis, we're going to see that we have time, because over time is how we're seeing this stuff changing. And then on our y-axis, we're going to have the number of individuals. So if we look here, we can see that the line ends right here, which means that this must be the highest point. And so since 70 minutes is my highest point, I know I have 14 little boxes. So that means I must be counting by 5. So each box equals 5. So I went ahead and I just labeled every 10. I'm doing this on a computer, so my numbers are kind of off a little bit, but they are every 10. And then I notice here that I level out at 700. So if I look in the data, I level out at 700. So this line must be 700. And if I count down, I see that there are 7 spaces, which mean that each box is equal to 100. So go ahead and you would graph that. And so you would get points for having correctly labeled your lines of the other 2 treatments. You have your axes labeled, including your units, as well as you have your labeled data lines. So here's what the student did. They have their lines graphed, they've labeled the ends of them, as well as they've labeled their axes. So they have full 3 points so far. So part B. As shown in the table, researchers established treatment group 2 with 5 individuals of each species, provide reasoning for reducing the initial population sizes. So we can see here that in group 1, we had 10. And then over here, when we looked at the separate container, we also had 10. So our third container, which may have both species A and B, we still wanted to have 10 so that we can have that control, that consistency of the same starting amount of individuals. So reduce the initial population sizes, keep the total number of organisms the same in all containers. Reduce initial population sizes, serve as a control for population density. So students said the students started with 5 individuals of each population to keep the starting total population at 10 individuals as in group 1. So the student has points there. Part C, the student claims that species A and B compete for the same food source. Provide 2 pieces of evidence from the data that support the student's claim. Now notice that they've asked for a specific number. Whenever they ask for a specific number, that means that the reader is only going to score your first 2 answers. So since they asked for 2, the first 2 are what is being scored. So you want to make sure that you spell this out for the reader. The first piece of evidence, the second piece of evidence, to ensure that your reader is looking at what you're considering your first and second piece. So if I look at this graph, I can see that if I look at species group A, species A group 1 and species A group 2, I can see that group 1 has a higher population density than group 2, being that since group 2 was being grown with another species, they were competing, so they had less of that growth of that population. I see the same thing here. I look at species group 1, species B, and then group 2, species B, I can see that group 1, of course, has a higher population density than group 2. And so that shows that there was competition. You could also talk about how it came down to the x-axis. So we ended up dying, or species B, group 2, died due to that loss of food source. And so here's the different answers you could have put on here. You could have talked about the growth rate is faster in 1, slower in 2, or that we grow to a higher population density in 1, or a lower population density in 2. Okay. So the student said, when both populations are in the same container, species A does not reach 1,500 individuals as it did in group 1. Species A only reached 1,400 individuals. Species B also did not reach the same number of individuals as it did in group 1. It reached 50 instead of 700 individuals. So part D, predict 2 factors that most likely limit the population growth of species A in treatment group 1. So we have to think to ourselves, like, what kinds of things are we looking at for carrying capacity? You don't just want to like tell them, oh, we've reached carrying capacity, or there's a lack of nutrients. Be specific. What is the thing that is causing the species to no longer be able to grow. Well, they're running out of food, they're running out of space. There's an increase in metabolic waste, and then there's a decrease in that dissolved oxygen. They don't have enough oxygen. Okay. So population growth is limited by the lack of enough food and lack of space. So there's your points there. And then part E, most protists contain an organelle called a contractile vacuole that pumps water out of the cell. The student repeated the experiment using a growth media with a lower solute concentration. Predict how the activity of the contractile vacuole will change under the new experimental conditions. Justify your prediction. So if the outside of the solution, right, our new growth media, had lower solute concentration, that means the solution is now hypotonic in comparison to the cell. And the cell would be hypertonic in comparison to this new environment. So since the environment is hypotonic, we know that water is going to move from hypotonic to hypertonic. So the water is going to move into the cell. If the water moves into the cell could potentially burst. But due to it having a contractile vacuole, it's going to pump that water back out. So I would predict that the activity is going to increase because I'm going to need to pump more water out as the water enters from the hypotonic environment to the hypertonic cell. And of course, contractile vacuole will be more active. Answer justification. The environment is hypotonic in respect to the cell. The cell is hypertonic in respect to the environment. Water is entered, or the cell has a lower water potential than the environment, or the environment has a higher water potential than the cell. So students said the activity of the contractile vacuole will increase because there will have become more hypertonic to the solution. Water from the media will come into the cell to try and create an isotonic media. So I hope that was helpful. Remember, 8 5 Hey, when's just the test. Bye, y'all.